Hello everyone, welcome back to Python for Bioinformatics. In the previous lesson, we learned how to split a sequence into smaller pieces using the split method. Today we are going to learn the opposite operation. What if we already have many sequence fragments and want to combine them back into one sequence? That's exactly what join method helps us do. Imagine these DNA fragments came from some earlier processing step. We have four separate pieces: ATG, GCT, AAC, and TGA. But suppose our goal is to reconstruct one continuous DNA sequence. We want the final result to look like this. How can we combine all these pieces efficiently instead of manually typing them together? Conceptually, this is a joining problem. We start with many fragments and want one sequence. This situation appears often in bioinformatics whenever sequence data is stored in separate pieces. And before we look at Python syntax, the important idea is simple. We need a way to connect multiple strings together. And Python provides us with the join method for this purpose. The general syntax is separator.join collection. Now let's understand this carefully. The separator is the character or string that Python will place between items while joining them. And collection is usually a list of strings that we want to combine. If the separator is an empty string, Python joins everything directly together. If the separator is a hyphen, Python inserts hyphen between items. And if it is a space, Python inserts spaces. The separator is what performs the joining. Let's see our first example. We create a list here called fragments containing 4 DNA pieces, and each item in the list is a string. Now look at the important line. The empty quotes here represent the separator. Since nothing is inside the quotes, the separator is an empty string. Python takes the list and combines every fragment without inserting anything between them. And the result is stored in a variable named as sequence. And finally, we print the result. The output is a single string. Python produced it by taking each fragment in order and connecting them directly together. Since the separator was empty, no extra characters were added. Biologically, we can think of this as reconstructing one longer DNA sequence from smaller fragments. Now let's change only one thing. The list is identical, but instead of an empty separator, we use a hyphen. And the syntax is still separator.join collection, and now the separator is hyphen. Therefore, when Python joins neighboring fragments, it inserts a hyphen between them, and the output becomes ATG-GCT-AAC-TGA. Notice that the hyphens were not originally present in the DNA fragments. Python inserted them because we specified a hyphen as a separator. This shows that the join method not only combines items but also controls what appears between them. Now let's look at another example. Here we have a list of codons, and each codon contains 3 nucleotides. And our goal is to create one continuous DNA sequence. Again, we use an empty string .join codons. So in this case, Python reads through the list and joins all codons together. and we get the output as one string containing every nucleotide from every codon. Python preserved the original order and combined all pieces into a continuous sequence. This is a realistic example of how sequence fragments can be merged into a larger biological sequence. A very common beginner mistake is writing fragments.join and within the parentheses we pass the separator. This is incorrect. The correct syntax, as we all know, is separator.join and within the parentheses we pass the fragments. This is because the separator performs the joining. The thing before dot join is always the separator, and the list of strings goes inside the parenthesis. A useful way to remember this is the separator is the object doing the work. An empty string joins without adding anything. A hyphen joins with hyphens, and a space joins with spaces. Whenever you are unsure about the syntax, think about what you want between the pieces, and that becomes your separator. Let's practice now. Pause the video and try solving this yourself. Here we have 3 DNA fragments AAA, TTT and CCC. Can you use join to create the sequence AAA, TTT, CCC? Pause here and give it a try. Now let's check the solution. We store the fragments in a list named as fragments, and then we write empty string dot join fragments. And we can see that Python created one continuous DNA sequence by connecting all 3 fragments without adding any extra characters. If your answer matched this, you have successfully used join. Now let's summarize today's lesson. Our problem was that we had many sequence fragments and wanted one continuous sequence, and Python gave us the join method as a solution for this. The key syntax is separator.join collection, and the version that we'll most often use in bioinformatics is an empty string.join sequence list, which combines sequence fragments without inserting any extra characters. Thanks for watching. Today we learned how to combine multiple sequence fragments into one sequence using the join method. And in the next lesson, we learn about removing unwanted characters from sequence data using the strip method. See you in the next video.